Okay, so welcome to this next video in the playlist on cancer. In this video what we're going to talk about is the hallmarks of cancer, which was basically a very famous review written by Hanahan and Weinberg in, I think, 2000, but they have since written modifications to it, i.e. released subsequent papers where they have improved upon their hallmarks of cancer. But in 2000, they wrote this very important paper, or review really, entitled "The Hallmarks of Cancer," and I'm going to outline some of the main hallmarks of cancer in this video. Okay, right. So, number one, the most obvious one, the one that most people will be able to give you, which is that cancerous cells have sustained cell growth. Okay, now what does this mean technically, sustained cell growth? Well, basically it means that they have the acquisition of oncogenes. Okay, so this means oncogenes technically. Right, so let's give an example of this basically. So, basically, a proto-oncogene now, let's talk about what a proto-oncogene is, because that's where oncogenes come from. So a proto-oncogene is a gene involved in the signaling involving cell growth, which, if you get a gain-of-function mutation in that gene, will lead to cancer. Okay, so let's say this gene here is a proto-oncogene. Let me just get my highlighters, we'll colour it in purple. Okay, so let's say this purple gene here is a proto-oncogene. Then, basically, it is a gene which, if you get a gain of function in this gene, then it will become what is known as an oncogene, and it will cause overexpression, well, it will cause overdivision of the cell, basically. So it will give you this property needed, one of these hallmarks of cancer. Okay, and the important thing is that we know, of course, that we have two copies of every gene. So let's say here's this other copy of this proto-oncogene. In order to get oncogenesis, in order for it to give you a characteristic, in order for it to produce sustained cell growth, you only need a gain-of-function mutation in one of the proto-oncogenes. That's important. So a proto-oncogene is a gene where you need, if you get a gain-of-function mutation in a single one of the copies of that gene that you have, it will lead to oncogenesis. It will take you part way there to getting cancer, basically. Okay, so GOF just stands for gain of function mutation. So if you get a gain of function mutation, then it's going to set you down the path of oncogenesis, basically. Okay, so gain of function is going to lead to oncogenesis. Right, which is just another name for the formation of cancer. Right, so now let's give an example of a proto-oncogene. A very famous example of a proto-oncogene would be the MYC gene. So again, you have two MYC genes, so we might call this the MYC gene maybe. And if you get a gain of function in MYC, then it will lead to too much MYC activity in the cell, and MYC is a very powerful transcription factor which causes the cell to move from the interphase of the cell cycle to G1 phase. So it's going to cause the growth of the cell. So this is a powerful mitogen, as it's called. It's actually often given the grandiose title of the cell's most powerful mitogen. So it's going to cause division of the cell. Okay, so MYC would be an example of a proto-oncogene, which if you get a gain of function mutation in just one of your genes for MYC, then that will become known as an oncogene, and it will set you upon the path to develop cancer, basically. Okay, so sustained cell growth is one of these hallmarks of cancer that, the hallmarks of cancer, how did I miss of there? The hallmarks of cancer, one of these hallmarks of cancer, and the technical details of what it means to have sustained cell growth is it means that you've got these oncogenes, you've got these gain of function mutations in the proto-oncogenes, and when you have a gain of function mutation in the proto-oncogene, that gene becomes known as an oncogene. Okay, so now let's have the second characteristic of cancer, the second hallmark of cancer, and this is the ability to bypass growth suppressors. Okay, so this specifically means loss of function in the tumor suppressor genes, basically. So there are other genes encoding for proteins which don't promote growth. This was about increasing the function of proteins which promote growth. This is about decreasing the function of proteins which suppress growth. And these proteins which suppress growth are known as tumor suppressors, and the genes which code for them are known as tumor suppressor genes. Okay, so perhaps the most famous example of a tumor suppressor gene would be the adenomatous polyposis coli gene, or APC, which encodes a protein which forms part of the beta-catenin destruction complex. So APC contributes to the beta-catenin destruction complex, which destroys beta-catenin, and in that way suppresses growth, since beta-catenin causes growth. So the beta-catenin destruction complex destroys beta-catenin and therefore prevents beta-catenin causing growth. So APC, this essential part of the beta-catenin destruction complex, therefore suppresses the growth of the cell. So it's a tumor suppressor gene. So in order to get over division, i.e. in order to cause oncogenesis, what you need is to lose function of these tumor suppressor genes. Now, again, you have two genes for these tumor suppressors, of which APC is an example. So here's your two genes of your tumor suppressor genes, of your tumor suppressor gene rather. Basically, will losing one be enough to cause loss of function of the protein? Potentially no, because if you knock out one, so let's say we get some sort of loss in function mutation in one, which means that either potentially the whole thing's deleted, or you've got some sort of sequence mutation in it, which means that it no longer encodes a functional protein, basically. Okay, so you've got some sort of loss of function mutation in one of them. The other one is still functional. What might happen then is you might get some sort of compensation. This one might up its expression so that it still produces, so basically it's going to produce twice as much as it would have done if this one was working, but it will take the buck, basically. It will upregulate how much it's synthesizing, and therefore the amount of the gene product of this tumor suppressor gene which you actually produce won't have gone down. Okay, so really to get complete loss of function of the APC or of any tumor suppressor gene, usually you have to lose both. There are some exceptions to that, but usually you have to lose both. So in order to go down the road of oncogenesis by targeting tumor suppressor genes, you have to lose both tumor suppressor genes so that protein that the tumor suppressor genes encodes for is completely, the function of it's completely gone, basically, so function lost. So you need two loss of function mutations, which are denoted LOF, for loss of function mutations in the gene, basically, in the tumor suppressor gene. So I'll just say tumor suppressor gene, TSG. Right, okay, so that's the second hallmark of cancer that Hanahan and Weinberg, and I just want to put their names somewhere so that if you want to go and read the review, you can do. Hanahan and Weinberg. Okay, so they're quite famous cancer biologists. Weinberg, I think it's Weinberg like that. Okay, right. So, We will continue our discussion of the Hanahan and Weinberg hallmarks of cancer in the next video.